compared to later in the semester when we look at plants and animals and fungi, Protista are not a very cohesive group. And this makes sense because remember what we said the other day, Protista is kind of the leftovers, the things that have eukaryotic cells, but they're not really united by anything other than having eukaryotic cells. So for everything else, what we're going to see is they're really a hodgepodge. And even though today we don't think of them as being classified into three groups, the ones that are related to plants, the ones that are related to animals, and the ones that are related to fungi. That's still not a bad way to think about them in the sense that as we discuss their characteristics, that's what we're going to see. So let's look at a cladogram, and this is not just showing Protista, although it's mostly Protista. Everything in the kind of the yellowish up here at the top is a group of Protista. Notice there are three groups that are not in the Protista. And that's the plants, which they call land plants. Some people do, some people don't. And the animals and the fungi. So when we look at these three groups, we'll consider them later in the semester. But for now, we're going to consider all these other things that, well, mostly they used to be Protista, but some of them were actually classified with plants or animals or fungi, depending on the situation. So when we look at Protista, I've purposely chosen a cladogram that's actually from the first edition of our book. And we'll talk about it, but there are two differences between this cladogram and the third cladogram, the third edition cladogram. Basically, these five groups up at the top are referred to as superclades. And our book, the current edition, divides Protista, or maybe I should say ex-Protista, into two distinct clades that have organisms we're familiar with. The Archaeplastida, remember chloroplasts are a type of plastids. The Archaeplastida include plants, and the Uniconta include animals and fungi. Now the other three groups, the Excavata, the Chromalveolata, and the Rhizaria, don't really have any groups that we're necessarily all that familiar with. If we look at Chromalveolata and Rhizaria, in our current edition of the book, these are combined into one group called the SAR clade. And we'll see why they have that abbreviation in just a moment. So this really shows 5 superclades. Our current book only shows 4. Otherwise, it's very similar. There are 1 or 2 other minor differences that I'll point out as we go along. But the reason I wanted to show you this version of the cladogram is that it shows how quickly the classification of eukaryotic organisms is changing. Plants and animals and fungi are pretty set. But when we look at eukaryotic organisms, that aren't plants or animals or fungi, there's a lot of debate about how they should be classified and where they fit in. So we want to keep that in mind. So, oops, went down a little too far. So let's look at these 4 big groups, the Excavata. If we look at the Excavata really have 2 things in common. they have an area that's excavated or kind of indented in the cell. They have kind of a hollow region. Keep in mind, almost all these organisms that we're talking about are single celled. So when I say they have a kind of an indentation in their cell, this is every cell. So you could think of this kind of like maybe a football or a basketball that's partway deflated. So it's got a little bit of an indentation in it. Now, they were originally proposed as a distinct group, not because of this hollowed out area, this excavated area, but they were originally proposed as a distinct group because if we look at the cytoskeleton in these three groups, the diplomonads, the parabasalids, and the euglenozoans, if we look at the cytoskeleton in these organisms, there are a few differences from the cytoskeletons of other eukaryotic organisms. Now let's think about the chrome alveolata and the rhizaria. And notice these are the two groups that I said are sometimes combined in the SAR superclade. Now, if we look at this superclade, oops, notice that there are two subgroups called the alveolates and the stramenopiles. So these are both part of the chrome alveolata. So if we think of stramenopiles, alveolates, and rhizaria, stramenopiles, alveolates, and rhizaria, this is our SAR clade. Now, if we look at these organisms, one of the things that we see is most of the members of this group, the stramenophiles, they have, many of them do, not all, but many of them have chloroplasts that were acquired by secondary endosymbiosis. So remember the other day when we talked about symbiosis, one of the things we said was that primary endosymbiosis, if we're talking about chloroplasts, would be when a eukaryotic cell ingested a cyanobacteria. And then secondary endosymbiosis is when another eukaryotic cell ingests this first eukaryotic cell that already has a cyanobacteria in it. So most of the members of this group have chloroplasts that they got through secondary endosymbiosis. Now, one of the things we'll see as we start looking at this group and some of the other groups, if you go back to your notes from the other day when we talked about, thinking of Protista as being separated into groups that resemble plants, groups that resemble fungi, and groups that resemble animals. Some of the Protista that resemble plants turn out to not be very closely related to each other. And the same thing for Protista that resemble fungi and Protista that resemble animals. So when we look at all these various organisms, we want to keep in mind that the old way of dividing things up was a lot easier to understand in the sense that everything resembled a fungus was in one group. But as we've been able to sequence DNA and as we've been able to analyze the amino acid sequence of proteins, it's turned out that things that carry out photosynthesis aren't necessarily closely related. Now, if we look at the rhizaria, one of the main things that they have in common is they all have pseudopodia, or I should say most of them have pseudopodia. And if we look at pseudopodia, remember those were little projections that amoeba put out to move. The rhizaria have pseudopodia that are called thread-like pseudopodia. And what this means is, think of a thread. Their pseudopodia are very, very narrow. And we're going to contrast that with some other organisms that have pseudopodia that have what are called tubular or lobe-shaped pseudopodia. The Archaeplastida archae, like archaebacteria or archaeology, archae means ancient. So these are believed to be the first group that captured a photosynthetic cell by endosymbiosis. And when we look at the Archaeplastida, notice that there's a subgroup within these called the green algae, which includes two groups, the chlorophytes and the charophyte. And then finally, we have the uniconta. And if we look at uniconta, if we go down a little bit, there's two main groups, amoebozoans, which literally means amoeba animals. And these are all organisms that, to some extent, resemble amoeba. And this is where we're going to find our organisms that have lobe-shaped or tube-shaped pseudopodia. And then we have the opisthecons, which include fungi, animals, and then the groups that are most closely related to fungi and animals. When we look at these five groups, most of these groups include some organisms that are parasitic. In fact, well, there's maybe one that doesn't. But most of these groups also include some species that are photosynthetic. There might be one that doesn't. Most of these groups include organisms that are heterotrophic, either by absorption or by ingestion. So remember, things that are photosynthetic are what we associate with plants. Things that are heterotrophic by ingestion is what we associate with animals. Things that are heterotrophic by absorption is what we associate with plants. So if we look at these four main groups, they almost all contain some things that are similar to plants, some things that are similar to fungi, some things that are similar to animals. So from that perspective, notice that's one of the reasons that that old system of classification is not so accurate. So what we're going to think about now is we're going to go through most of these subgroups, and we're going to look at a photograph, well, a few diagrams, but mostly photographs of specimens in each of these groups. And one of the things that you'll notice as we go through these photographs, now this is something we would normally do in lab, but one of the things you'll notice is that a lot of these organisms look very, very similar. Well, remember we said when you're studying the anatomy of something, anatomy is something called morphology. Morphology is the different parts, what they look like, how they fit together. And if we look at how Protista were classified before we had access to DNA analysis and protein analysis, all biologists had to go on was morphology, what the organisms look like. And a lot of these organisms are single-celled organisms. So, you know, we're even talking about organelles inside the cell when we talk about morphology. And depending what type of microscope you have, you might not be able to see every organelle that's in the cell. And in most cases, when you stain cells so you can see the organelles, the stain also kills the cells. So the good news is you can see the parts. The bad news is once you stain them, you can't see how the organelles function, what they actually do, because the cell is dead. So it's always good to think about history and put it in perspective. So today we might look at dividing Protista into things that resemble plants, organisms that resemble fungi, and things that resemble animals. We might look at that as being a very naive approach. But at the time, that was really all biologists had to go on. So when we look at these organisms, we want to keep that in mind. So we're going to go through the groups one by one. We're going to look at some photographs of slides. Some of them I've gotten from the internet. Some of them I've gotten from people who have taken photographs. Some of them I've taken photographs of. So the first group we're going to talk about is diplomonads. And keep in mind, we're really starting on the far left of that chart up above. So if we look, we're starting over here with diplomonads, and we're going to work our way over to the right. So if we look at diplomonads, they are a clade within the superclade excavata. So remember what we said, they have an excavated area. Well, in this photograph, it's very easy to see. You should be able to kind of visualize the three-dimensionality. They have this groove down here at the bottom that's kind of shaped like an upside down U, and it leads underneath this structure up here. So this is the excavated area in the cell. Now, this is a single-celled organism. It's magnified quite a bit. And if we look at the diplomonads, one of the ways that they got their names, di means two, is that all the cells in this group have two haploid nuclei. Now remember, haploid nuclei have the 1n number of chromosomes. This is what we usually associate with, if we're thinking about a person, this is what we associate with an egg cell or a sperm cell. Most of the cells of our body are diploid. But notice for these organisms, the cell that makes up their body is haploid. So again, I think I mentioned before, but one of our main themes this semester is we have this mistaken notion that all living organisms have a biology similar to ours. There are a lot of living organisms we're going to encounter this semester that are haploid, not diploid. If we look at diplomonads, they appear to lack lysosomes and peroxisomes and a Golgi apparatus. Now, notice I'm saying they appear to lack these structures. We're pretty sure they don't have them, but the reason we want to say that is because it turns out that they have modified mitochondria, and these modified mitochondria are called mitosomes. They're very reduced in size and function compared to a mitochondrion that we would see in most eukaryotic cells. Remember, in most eukaryotic cells, we have the Krebs cycle and we have the electron transport system, the aerobic part of aerobic cell respiration taking place in the mitochondrion. What's left of their mitochondria don't function in aerobic cell respiration. They actually play a role in oxidizing sulfur, typically. But if you would have taken this class, say, 20 years ago, I would have told you that diplomonads don't have mitochondria, period. Because until we could do some genetic analysis and until we found this organelle in the cytoplasm and were able to analyze it and realize that it does have some genetic material in it, and the genetic material is similar to what we find in mitochondria, we just thought they didn't have mitochondria. But it turns out their mitochondria over evolutionary time have kind of deteriorated and adapted to be used for different functions. So that's why I say they appear to lack lysosomes, peroxisomes, and Golgi apparatus. As far as we can tell right now today, they don't have those things. But 20 years ago, we thought they didn't have mitochondria. So maybe there's some trace of those organelles left, and we just haven't figured it out yet. The group that's closely related to that, notice the group that it's split off from most recently. Remember how we determine how closely related organisms are according to cladistics. The group that's closely related to that is the parabacillids. And they move by flagella. In fact, almost all the organisms in this big group, the excavata, move by flagella. There are a few weirdos, but there are in every group of Protista. And they have several different clusters of flagella. So we can't really say that they all have 8 flagella, because when we look at These guys, they have 2 nuclei, and each nuclei is in charge of 4 flagella. So there's a total of 8 per cell. These organisms also don't have true mitochondria. So again, 20 years ago, if we were talking about these, we would have said they don't have mitochondria, period. But it turned out they have the remnants of some mitochondria. Their mitochondria are called hydrogenosomes. And again, they have a metabolic role, but they don't really work in aerobic cell respiration like we think of mitochondria as doing. They do have Golgi apparatuses. And in fact, their Golgi apparatuses connect to the basal body of their flagella. Remember, the basal body is what's at the very bottom of a flagellum or a cilium, where it connects to the cell. And the connection is by something called parabasal fibers. Well, this group is parabasalids. So this is really what gives this group its name. Now, parabasalids turn out to be very important ecologically because a lot of them contain cellulose digesting bacteria. So these cellulose digesting bacteria live in, I said their digestive system, but it's really just inside the cells. They don't have a true digestive system because it's one cell. And then these guys live in the digestive system of termites and cockroaches. Well, termites and cockroaches can eat wood, and wood is mostly cellulose. But there's a problem. Termites, cockroaches, people, there is no living animal that contains an enzyme called cellulase. Cellulase is the enzyme that breaks down cellulose. So if you want to get nutrition from eating wood, you have to have cellulase. So animals don't make cellulase. So what's the alternative? Animals can contain bacteria that digest cellulase. And in the case of termites and cockroaches, they have these parabasalids in their digestive system, and the parabasalids contain bacteria. And the bacteria digest the cellulose. And when they digest the cellulose, they ultimately break it down into monosaccharides and disaccharides. And they get so much wood by living inside these termites and cockroaches that there's an excess of monosaccharides and disaccharides. So what do? They just release those into the digestive system of the termite or the cockroach. And then the termite or cockroach can absorb those carbohydrates and use them for glycolysis and other purposes. Notice again, this is an example of something we talked about the other day, symbiosis, where organisms have an arrangement where they benefit by living with another organism. In this case, again, what does the Protista get? Well, if it's inside a termite or a cockroach, that termite or cockroach is a lot bigger than this parabasalid is, so it gets protection from some predators. What does the termite or cockroach get? They can extract nutrients from wood, or they can at least get some nutrients from wood. They don't really do the extraction. That's what the parabasalid does. Now, the biggest group that's part of the excavata is a group called the euglenozoans. And anytime you see zo at the end of a word or the beginning, zo, like zoology, means animals. Think of a zoo. What's in a zoo? Animals. So these guys are called euglenozoans, but notice there are two groups. One is the euglenids, and almost all the euglenids have two flagella. Remember what we said about this group. They generally have flagella. That's how they move around. The euglenids usually have two. And even though this whole group is called the euglenozoans, there are a lot of photosynthetic species in the euglenids. Now, notice this also shows how their are a lot of cellular organelles that we don't find in human cells. Euglenids, and these are still, all these organisms that we've talked about so far are single-celled organisms. Euglenids have an eye spot. They have a small organelle inside the cell that can detect light. Now, they don't see images, they can just detect light. So you might think, well, if they can only detect images, if they can only detect light, if they can't see images, you know, what advantage could that be? But since a lot of these species are photosynthetic, they want to move towards light. So when they detect light, they move in that direction because obviously you need light to carry out photosynthesis. The other group is called the kinetoplastids. And the kinetoplastids include a lot of parasitic species. One of the most famous is an organism called Trypanosoma. So if we look here, this is a slide of human blood. And if we look at these kind of roundish orange cells, these are red blood cells, also called erythrocytes. If we look at these little squiggly things in between them, this is Trypanosoma. And Trypanosoma, if we look at some of them, if we look up here, you can see that part of this little squiggle is thicker. That's where the cytoplasm and the nucleus, this is again a single cell, that's where the cytoplasm and the nucleus is located. And then this skinnier squiggle down here is a flagellum. So when Trypanosoma gets into your blood, it actually swims around in between your blood cells. And it attacks blood cells and can partially eat them and destroy them. Some of these blood cells that are kind of the darker color are cells that have been at least partially eaten by Trypanosoma. Now, Trypanosoma is a good example of how evolution has produced organisms that use all sorts of strategies to survive. Remember when we talked about viruses and bacteria the other day, and we were talking about how your immune system detects viruses and bacteria. And we said it mostly detects them by proteins. In the case of a virus that are in that viral envelope or in the protein capsid. And we said if those proteins change, like the flu virus usually mutates once or twice a year, so that's why you have to get a new flu vaccine next year, because the flu vaccine teaches your immune system to recognize the proteins on the flu virus that's around this year, but after the genetic material mutates next year, the genetic material is the instruction for the proteins. So the virus makes different proteins, so you need a new vaccination. If we look at Trypanosoma, Trypanosoma has a very large genome. It has lots and lots and lots of genes. And these genes can make proteins that can be combined in different ways. You could think of these proteins maybe as being like letters of the alphabet, you know. If you have 3 letters, CAT, you could make cat, but you could make act. So here what we see is after Trypanosoma has been in your blood for a while, it starts removing proteins from the outer part of the cell, and it uses its DNA to make new proteins that it puts in the outer part of the cell. So what does this strategy do? Well, if your immune system encounters Trypanosoma and it sees certain proteins, now those immune system cells in your body are going to travel through the body looking for that same protein. But in the meantime, what happens? Trypanosoma are taking that protein out of the cell and replacing it with different proteins. And, you know, might seem like your immune system should be smarter, but it's not. If it doesn't find the precise protein, or in some cases, combination of proteins that it's looking for, it doesn't recognize trypanosoma and it leaves it alone. So trypanosoma is very good at evading your immune system by changing its proteins. So again, you know, big part of the theme of this class is we don't want to think human biology is necessarily the way that everything else works. Okay, so the next big group is this SAR group, the Stramenophiles, the 3 over here, the alveolates, and the rhizaria. So, oops. So let's think about these organisms. So as I mentioned, a lot of these Stramenophiles have chloroplasts that they've gotten a hold of by secondary endosymbiosis. So remember, one of the things we said, usually if cells get a chloroplast by endosymbiosis, there are 2 membranes around it, primary endosymbiosis. If they get a chloroplast by secondary endosymbiosis, there are sometimes 3 or 4 membranes around it. Not always, sometimes still 2, but so diatoms are a group of Stramenophiles. They're photosynthetic, and they have a wall that's basically made of the same material that glass is, something called silicon dioxide. And this glass is contained in an organic matrix. But when we look at these, what we see is there's a lot of artwork that's kind of been done with diatoms. These are some of their shells. And so you can see their shells sometimes, you know, look artistic. With some of the shells, it's easy to see, like here, you can see some big holes in this third one from the right on the top. You can see some holes here, but all of these shells have little holes in them, and water can flow into these holes. Now, these shells are unique in living organisms. If you would look at the shells that, say, a clam makes, the 2 shells in a clam are basically mirror images of one another. They're not exactly perfect, but they're pretty close. But if we look at the shells in these organisms, if we focus on this one, the third one in the top row, you might be able to see this kind of darker ring just to the right of all those holes. This darker ring, this is one half of the shell, and then this lighter part down to the right, or over to the right, I should say, not down. This lighter part is the bottom half of the shell. What's unique about these shells is they're very asymmetrical. One part of the shell is usually much larger than the other, and the part that's smaller kind of fits over the larger part, kind of like a lid would go on a box. And this is very unusual when we look at shells in living organisms. They're almost always very close to being equal. Now, there are huge numbers of diatoms in ocean water. If you would take a bucket down to the beach and you would scoop up some seawater, there's several million diatoms in that bucket. So there's tons and tons of these. Since they're photosynthetic, this means that they're very important because organisms like ourselves, or in the ocean, fish that are heterotrophic, have to eat things that are photosynthetic. So they're very important producers in the ecosystems of the ocean. And they make up a large portion of what's called the plankton in the ocean. Plankton comes from a Greek word that means wander. And what we find in the oceans is that there are millions and millions of plankton, and they're relatively small organisms. Now, diatoms are microscopic, but some plankton are actually big enough you can see them without a microscope. But the key is all these plankton are so small that they really can't fight the current in the water. So this is why they're wanderers. They basically drift on the currents, they drift on the tides, they follow the direction that the water is going. But diatoms are very common. Now, because of their structure, think of those shells. Because of that glass structure, this means that we've got glass with basically little holes in it. After diatoms die, we can dig up their skeletons, if you will, and their skeletons basically are glass with little holes in it. So there's something called diatomaceous earth, earth that we dig up that has a lot of diatoms in it. And it's often used to make filters for things like swimming pools, because water can go through these little holes, but we can filter out. Keep in mind, these are single-celled organisms. So we can filter out particles that are very small. So they're very useful for making filters. You do sometimes have to be careful walking on diatomaceous earth, because this shell is glass-like material, so it can scratch your feet up. Now they're small enough, it's not going to be a big gash, it's not going to be a huge cut, but that is a potential problem. Diatoms are very closely related to brown algae. And notice the Protista that we've been talking about so far have been organisms that are single-celled. One cell makes up their body. They're sometimes called unicellular. Uni means one. Brown algae are multicellular. And these are the largest of all the organisms that used to be considered protists. They can reach lengths of up to 30 meters. And 30 meters is about 100 feet. Like most protists, they tend to live in water. These are often used in movies. This is an actual photo of a scuba diver swimming among some kelp. And kelp is a type of brown algae. And whenever you see a movie where there are some scenes shot underwater and it looks like the scuba diver is swimming through these underwater jungles, that's all brown algae. Those are the only type of plants that live at depths more than about 10 meters, about 30 feet. The only type of big plants, I should say. We have a lot of these small photosynthesizing organisms. In fact, if we look at brown algae, they're so large, they're considered to have organs. Mostly we find organs when we look at plants and animals. So if we look at brown algae, one of them is a species called Macrocystis pyrifera. Brown algae have a holdfast. You could think of the holdfast kind of like the roots of a plant. Except all the holdfast really does is anchor this brown algae. So it might be dug into the sediments at the bottom of the ocean. It could be attached to a rock or to a board that's sunk. Now, keep in mind, there is one significant difference. The holdfast just attaches the brown algae. If we look at a plant, the roots attach a plant and anchor it in place, but they also absorb nutrients from the soil. The holdfast doesn't absorb any nutrients. Everything above the holdfast is called the frond. And there are three main parts to the frond. There's something called a stipe, and the stipe may branch. And the stipe is basically like the stem of a plant. There are blades, and the blades are similar to leaves that we would see on a plant. Something that plants don't have is in the stipe at various locations, there are gas bladders. And what this organism does is it pumps gas from the water into this gas bladder. So this gas bladder works kind of like an inner tube at the swimming pool. This gas bladder keeps the plant afloat so that it can stay up, because keep in mind, This might be attached 60 feet under the water. And the deeper you go underwater, you probably know this if you've ever, been at a lake or something and tried to dive down as far as you could dive, the water gets darker. Well, if you're brown algae photosynthesize. So if the water gets too dark that not enough light comes in, it's difficult to photosynthesize. So for that reason, these gas bladders are important. Now, keep in mind that like a lot of algae, brown kelp is actually used for food in many places. Brown kelp is considered to be healthy for you in the sense that there are a lot of minerals dissolved in seawater, and the kelp takes some of these minerals out of the seawater and incorporate them into their cells. However, notice I do have some bad news for you if you're one of those people who really like sea salt. Sea salt is pretty much sodium chloride, which is the same thing that regular table salt is. So there's really no evidence that sea salt is healthier for you, either in terms of containing a lot of minerals or in terms of being good for lowering your blood pressure. If something has sea salt on it, that's the same as having salt on it. Now let's look at the next group, the alveolates. And the alveolates have membrane-bound sacs called alveoli that are right inside the plasma membrane. These membrane-bound sacs are actually a modified type of vesicle. So you might remember vesicles from 105 carrying proteins from the endoplasmic reticulum to the Golgi apparatus, or carrying them from the Golgi apparatus to their final location. And remember, some of those vesicles became peroxisomes and lysosomes. So if we look at the alveolates, they've got these structures. Now, is there anybody who's had anatomy who knows what alveoli are in anatomy? Is there anybody who can tell me that? Well, in anatomy, alveoli are little structures. They're kind of shaped like grapes, very, very, very tiny grapes. But they're little kind of circular, or I should say spherical, they're three-dimensional, spherical structures in the lungs. And this is where gases are exchanged. Oxygen enters your blood, carbon dioxide leaves your blood, and it does this across the plasma membrane of the cells in the alveoli. The reason I bring that up is this is a good example of how the same word is often used in several different places in biology. So when we see this, we don't want to think of the human lungs. So let's look at one group of these organisms, and then I think we're going to be about out of time. Let's look at the dinoflagellates. Now, if you look at the name of these organisms, you should be able to tell that they're going to move by flagella. And if we look at these organisms, they have a pellicle right inside the plasma membrane. But if we look outside the plasma membrane, they have a series of cellulose plates. And you could think of these cellulose plates as being kind of like the armor that ancient knights used to wear. Most locations, the cellulose plates overlap, so they do provide some protection for the cell. But there are two places where there are definite gaps between the plates. And in these gaps, these little grooves, there are flagella that sit. And there's actually a slight difference in structure between the two flagella. So there's a transverse flagellum and a longitudinal flagellum. And we can tell these two flagella apart if we look at their microscopic structure. Now, there are some dinoflagellates that are photosynthetic, but when they've been studied more carefully, it turns out a lot of these species are actually what we call mixotrophic. Now, organisms that are photosynthetic, we sometimes call autotrophic. Auto means self, trophic means feeding. So plants, anything that's photosynthetic is autotrophic because they make their own nutrients. They make carbohydrates and proteins and lipids through that process that we talked about, the Calvin cycle, the light independent reactions. Things that are heterotrophic, like people, we have to get our nutrients by eating other organisms. We could eat a plant, or we could eat an animal, we could eat algae, but we have to get our organic molecules pre-made. We can modify organic molecules, but we can't make organic molecules from inorganic starting points, like carbon dioxide and water. Things that are mixotrophic do both. So a lot of these organisms carry out photosynthesis, but if they happen to bump into a smaller cell, they also eat it. So they have some characteristics of plants, some characteristics of animals. And again, this is one of the reasons why Protista are so hard to classify. We don't have any animals that carry out photosynthesis if they can't find food, but these organisms can do both. Now, these organisms contain a red photosynthetic pigment. The dinoflagellates are the group that we talked about the other day that were originally called the fire red algae. So if we have a population explosion of dinoflagellates, this creates something called a red tide. Because there are so many of them, the water looks like it's red. And this is a problem because dinoflagellates produce a neurotoxin, and neurotoxins are poisonous to your nervous system. These neurotoxins are only poisonous to vertebrates. So when there's a red tide, a lot of times we have a big fish die-off. You'll have hundreds or thousands of fish that eat some of these microscopic organisms in the seawater, and they'll be killed by the neurotoxin. And for people who live on the coast, when there's a red tide, states actually issue a warning and tell people they shouldn't be eating any oysters or clams, any shellfish, because notice that this neurotoxin is only poisonous to vertebrates. So shellfish, like clams and oysters, can eat these microscopic organisms. They're not poisoned by the neurotoxin, but they store it. So if you would eat these shellfish after that, you get what could be potentially a fatal dose of the neurotoxin. We don't hear much about it in Chicago because the types of dinoflagellates that produce this neurotoxin are found in marine water, oceans. They're not found in fresh water. So we're not going to have that become an issue in Lake Michigan. At least not right now. It could happen at some point in the future. Are there any questions for now about the groups of Protista that we've talked about? Okay, if not, I'm going to leave this screen. Don't forget, evolution questions are due tonight before midnight. And if anybody has any questions, we'll see if we can answer those now. But if not, I'll let you go and wish you a pleasant weekend. Go ahead, Nick. Trypanosoma is not a word, a worm, someone asked. All worms are multicellular animals. So it does have kind of a worm-like body shape, but actually it's a single-celled organism. Now, there are some worms that are multicellular animals that are actually not much bigger than Trypanosoma is, but Trypanosoma is a Protista. It's a unicellular organism.
Oh, there we go. Can you hear me?
Yeah, I'm sorry. I was at a meeting last night and they did that to everybody at the meeting, and I guess it changed my preferences.
Oh, no worries. I have a question about question 26 on the answer set there, or the question set.
Okay. Can you read it to me? I don't have it up right now, and then I won't have to pull it up. Otherwise, I'll pull it up.
Sure. Which level of classification is based on the ability of organisms to reproduce together?
Mm-hmm.
Oh, my question might give away.
Well, so think about which of those groups includes organisms that could theoretically reproduce together, at least if we're looking at organisms like ourselves. One of the things we talked about was, bacteria never reproduce with another organism, so that's kind of meaningless when we apply it to them.
Oh, I think I might have been overthinking it then, because all the answers were examples of levels of classifications, but not the.
Yeah, so this would be, okay, can all the organisms in a domain reproduce together? What about all the organisms in a phylum,
Okay.
Any other questions?
Do we have class on Monday?
Is Monday President's Day?
Okay, never mind.
I'm not sure. I was asking.
I think so.
If it's President's Day, we do have the day off. I knew it was either this week or next week, and I didn't pay attention. But if it's President's Day, we don't have class, so we won't see each other till Wednesday, actually.
Thank you. Have a nice day.
You too. Bye-bye.
Have a good weekend.
You too. I'll see you Wednesday.
See you Wednesday.
I have a quick question.
Sure.
For the evolution questions, is it okay to write the answers on a separate sheet of paper? Or did you want us to type it?
It's probably easiest if you just type it, but if you do them on a separate sheet of paper, that's okay. The main advantage is if you do it directly on the test, then if you decide you want to get your test back, I can send it back to you and you'll have your questions and your answers on the same page.
Oh, okay. Okay, thank you. Have a good weekend.
You're welcome. But if you decide you want to do them on a separate paper, no problem. Just make sure it's some kind of text document. So it could be a Word document or Pages or TXT or RTF, or I'm probably leaving out some file type, but.
Okay. Thank you.
You're welcome. Have a good weekend.
You too.
Okay, see you Wednesday.
Bye-bye. See you Wednesday.
Mr. Griffith?
Mm-hmm.
Yeah, I have some questions I want to ask concerning the homework. Are you going to be free today?
We can meet later today, or if you're free now and you want to stay on, it looks like everybody else is gone. You can just stay on now.
Okay. So I was doing some calculations on the Adiwemba question.
Mm-hmm.
And then I tried the other way around, but I didn't get the same answer.
I'm not sure what you mean by the other way around.
Okay. So I did this calculation. Okay.
Do you want me to let you share the screen so you can show me what you did, or?
Yes. Okay.
Okay, it may take a minute, but you should be able to put up your screen now.
I think I'm sharing now.
Yeah, I can see your document.
Okay, so yes. Okay, so I think the B, was it B? I think the A says what frequency of Huntington's disease in Barranquitas?
Right, so we're talking about that particular town.
Okay, we're talking about so we're talking about the dominant trait, right? The one for dominant, the capital H.
So it looks like in this city, you know, it's 411 people out of a little over 4,000 that have the disease.
Yes. Okay.
And since this is phenotype, you know, we could take the total population size and we could subtract. Everybody who doesn't have the disease must be normal.
Right.
So we could, so the frequency of normal is going to be about 3,600 divided by the total population.
Okay, yeah.
You see how I'm figuring that out?
Yeah. Come on, let me use my calculator. I want to, because I did something and I was trying to get the decimal, and then I was getting two different answers when I did it in another way. So
Now, the only thing is make sure you show all your work in terms of, you know, the formula too, I said. Okay, it looks like you got it.
and so the C says something like, what is the frequency, the allele frequency of H, which is supposed to be the normal phenotype, right? The allele frequency for the normal phenotype.
Right, that's the allele that gives you the normal phenotype. So you would take whatever your answer is for B, and you would take the square root of that. So your answer looks about right. I'm just estimating. I'm not putting it into a calculator, but that looks like that's pretty close to right.
Yeah, I think this is the D. This is where I got it. So when it said what is the allele frequency of H, so when I took the square root of 0.1.
Well, but 0.1, is that the dominant or recessive? Your 0.1.
I think 0.1 is supposed to be the dominant, right?
So remember, you can't take the square root of the dominant. You can only take the square root of the recessive, because remember what we said, if you take the square root of the dominant, all those people aren't homozygous dominant. Some of them are heterozygous. You can only take the square root of the recessive phenotype, because everybody who has the recessive phenotype has to be homozygous recessive. And then the dominant is always going to be 1 minus whatever you got for the recessive allele. So you can find the dominant phenotype, because that's what we look at. That's what we see. You can find that without knowing the recessive phenotype. but you can't find the dominant allele frequency without knowing the recessive allele frequency. Because the only way to find the dominant allele is 1 minus whatever the recessive is for the problem.
I was getting it though. When
I
I
mean,
did
it
it like
looks-
this,
It
when I did like this,
looks,
0.05.
I was gonna say, it looks like what you have. I up through D is correct. So I mean, then it's just kind of a matter of, using the right formula and plugging in the correct numbers.
Right.
Yeah, I think maybe so you're confusing the genotype and phenotype, because phenotype, we can find the dominant phenotype because we can look at individuals and see if they have this disease or not. We can't find the dominant allele by just taking the square root of that though, because they're not all homozygous dominant. Some of them are heterozygous. And when you take the square root mathematically, that's kind of making the assumption that everybody is homozygous, whatever allele you're looking at. So it looks to me like you have the right answer through D. So if you keep, using the correct number and putting those answers in that you've gotten for A, B, C, and D, depending on what the question is.
Yeah. So the last one was like the H. I think it was the H. How many people carry at least one recessive allele? So
Think about who has a recessive allele out of there's 3 genotypes, right?
Yes.
There's big H, big H, big H, little h, and little h, little h. So who carries a recessive allele? Well, people who are heterozygous carry a recessive allele. People who are homozygous recessive, well, carry 2 recessive alleles. So this means that, think about our Hardy-Weinberg problem, we're going to have to add 2 times big H times little h plus little h squared. Now that's just the frequency though. 2 times big H little h plus little h squared. So since it says how many people, what do we have to do whenever it says how many people?
Multiply by the population size.
Yeah. So after you add those frequencies together, then you're going to have to multiply by the total population size.
So we add first.
I'm sorry, what?
I said we add first, right? So
yeah, add those 2 frequencies first. I mean, mathematically, you should be able to take 2 big H little h and multiply it by the population size, and then little h squared and multiply it by the population size. And you could add those if you wanted.
Yeah. So what I use another, can I use another way? Because what I did here was I looked at the dominant, like the question says how many people carry at least one recessive, right? And then when I got the value for the people who are, I think I got 1915, they are homozygous dominant. That means they have small
Big H.
Okay, homozygous, yeah, big H. And then the question says carry at least one recessive. So I just removed it from the population size.
Yeah, you could do that too, because everybody who's left,
Right.
since they have 2 alleles and they're not both big H, they've got to have at least 1 recessive. Yeah, that should work too.
That's what I did. This is just, yes. Done with that.
So I think you were on the right track. You just didn't realize you were on the right track.
Right. I got confused when I got confused when I did was it the H square? I was looking at this formula, P square plus 2PQ